much knowledge, but it will help you, obviously, if you do have knowledge from before. But it also might be a disadvantage if you have some knowledge before that's incorrect. So we'll address this together. All right, so let me open my PowerPoint first. Get a little organized here. Is this what I want? Yes, I think so. Okay. So, can you see that? You can't see that. I haven't shared it with you yet. All right, good. Okay, here we go. Yes? Everyone can see Lita, right? All right. All right, let's hide that, and then we'll go to here. Okay. And I'm going to slideshow it. Play from the current slide. Okay. So today we're going to start genetics. Genetics is interesting. It's kind of difficult, though, if you don't really get familiar with the terms that we're going to use. So just really try to embrace it and use the terms that we're going to use today in the correct manner. Okay. So the agenda for today, we're first going to review mitosis. Oh no, we're not going to review mitosis. Just kidding. We're going to introduce Mendelian genetics, and then we're going to practice these monohybrid crosses, which follow the rules of Mendelian genetics, right? All right. So, have you guys ever heard of Gregor Mendel before? Yes? No?
Yes.
All right. Well, so he was I think he was it Austria? He was a monk, right? You guys all know what a monk is, right? So someone who lives in a monastery who, they live a very Spartan life. So they're not really concerned with worldly types of stuff. They have very empty sort of self-denying sort of lifestyle, right? But in any case, he was really interested in this garden that they had at the monastery, right? So he did a lot of experiments of crossing different plants and then seeing what offspring plants they had, right? At the time when Gregor Mendel was doing his work, they didn't know DNA, and they didn't know that DNA was the carrier of genes, which was the responsible for carrying on these traits. But so he just really studied all these patterns, which made it even more impressive because he just kept really good records of all these different crosses that he did, but he didn't actually know that the carrier was DNA or that it was carried in these things called genes. He called them inheritance. I forget what it was. It was inheritance units or something that he didn't call it genes. Okay, so his nickname was the father of genetics because he did sort of establish the main rules of how genes are passed on from parents to offspring, right? So he studied, the main thing that he studied were these pea plants called Pisum sativum, right, which were just regular old garden peas, right? So this was the organism, his organism of choice that he worked with, right? And I know when we talked about mitosis and meiosis, we talked a lot about humans and how humans create their gametes, but it should also be emphasized that plants also reproduce sexually, right? Meaning that they produce egg and sperm variations that then unite to form a zygote and then a offspring, right? Okay, so you guys know who Gregor Mendel is. He's the father of genetics, right? Okay, so when we talk about traits, it means a specific visible variation of some sort of aspect. So for example, seed shape, you see that at the top? That's the trait. And there are two versions of that trait. You could have a round seed shape or you could have a wrinkled seed shape, right? For seed color, you could have a yellow one or you could have a green one, right? And those two happen to be round as well, right? For flower color, you could have purple or white. For pod shape, you could have that kind of puffy, inflated one, or you can have the constricted one where you can see each individual outline of each pea. You can have the pod color, right, which could be yellow or green. And then here's another trait, flower position. You notice how the flowers here are lower, whereas terminal means on the top, at the very extremities of the plant, right? And then stem height as well, look how short this one is. So this is a dwarf, and then this one is the tall version, right? So you guys can all recognize that there are usually two or possibly more variations of traits, right? Okay. Okay, so we need to sort of talk about these definitions, right? So we need to first talk about the word an allele, right? Do you guys remember that? What are homologous chromosomes again? Do you guys remember what homologous chromosome is?
They were chromosomes that carried information about the same trait, but they were not necessarily like identical.
Exactly. Good. So they're two chromosomes that, like, on a specific, like, designated part of that chromosome, they have like a specific gene which is responsible for a certain trait. So let's call it, like, suppose we have, like, on these chromosomes, which are like kind of, you know, that X kind of shape. At one part, there's like a band that like represents a gene that maybe stands for let's say like color, right? Like, let's say seed color. Okay? And then maybe one version of that gene is green, and then on the homologous chromosome, let's say, that same gene for seed color is present, but instead of being green, it's yellow, right? So the green version and the yellow version, their relationship to each other is known as alleles, right? So they're basically two variants of like the same gene or the same trait, okay? So they're versions of a gene that are usually, in genetics, you represent them by a single letter, right? Either a capital or a lowercase letter, but it's like one single letter, right? In one gamete, right? Think about this. In one gamete, so if a plant is producing like eggs and sperms, right? In one of those gametes, how many alleles for a specific gene will be present? Because we said during meiosis, which creates the gametes, the homologous chromosomes, the ones that are homologous to each other, they separate in meiosis 1, right? In anaphase 1 of meiosis 1. So that means that the two alleles, which are on separate homologous chromosomes, right? What happens to them then when they get made into gametes? How many alleles get put into each gamete? One for every single trait, right? So for seed color, let's say, I'll only have one, in a given gamete, I'll only have one of the alleles for that given trait, right? So you guys have to remember that. So if I give you like a P, or if I give you like a single P here, you'll know that that's only referring to like one gamete. Not, you know, if it has only one of them, only one copy, that means it's a gamete, right? Okay, so an allele is like the version, it could be one or the other, right? Okay, so how many alleles does each individual have for a given gene? So if you're an individual, you're a full organism, right? How many alleles will you get for a given gene? Anyone?
Two.
All right. Two. Good. You get two, right? One from your mom, right? And one from your dad. So every individual will have two alleles for any given gene, okay? Are you following me? Yes? Okay. All right, so the genotype is basically the combination of alleles that you get for a specific gene, right? So for example, like we said that each individual has two alleles for a given gene, one from their mom and one from their dad. So that is that individual's genotype, right? So for example, like suppose I have like purple flowers, right? That means that maybe I got this, suppose I got this purple flower allele from my dad, and I got this purple allele from my mom. So this makes up. Is everything okay there? I hear a lot of like festive music. Who was that? No takers. Okay. So also I could have gotten like a small allele. Like, so I could have gotten this allele from one parent and then this allele from another parent. But this is like my ultimate, like, you know, genetic makeup. This is my genotype for that given trait. Okay. So you guys understand the difference between a genotype and an allele, right? An allele is like what goes into a gamete. Like one allele will go into each gamete, right? whereas like two alleles make up a person who is like an individual. Okay? All right. So homozygous, homo means what?
Same.
Same.
Homo means same, right? So if you're homozygous, that means you have two alleles that are the same. So it could be capital P, or lowercase p, right? So these two alleles are the same, and these two alleles are the same. If they're not the same, so it's not going to be homo, but it's going to be called heterozygous, right? Which means that you have one of the capital one, and then one of the lowercase one, right? So remember, a genotype is two letters, an allele is just a single letter. Okay? Homozygous or true breeding, you have to know this term too, means that they have the same two alleles, whereas heterozygous means they have one of each allele. Okay? Do you guys, so phenotype refers to, it doesn't refer to like, what alleles you have, it refers to what you look like. So this is like your physical. I always remember phenotype, like physical. So it actually shows like what you actually look like. So it's not going to be represented in letters. It's represented in like a description, like purple, white, wrinkled, tall, or whatever, right? Okay, questions so far? Okay. I'm going to move on. Okay. So Mendel had 3 different laws of inheritance, and these are, I think they're pretty straightforward, but we just have to make sure we understand them. So the first one was called the law of segregation, meaning that suppose you're a parent, right? What's your genotype? What's in a, like, let's suppose we're talking about the gene for flower color. It could be either purple or white. Suppose you're a parent and you're homozygous dominant, meaning you're homozygous for the purple allele. That means what's your genotype?
PP.
If you're homozygous, that means 2 of the same. And for the purple allele, which is the capital P, so what's your genotype going to be?
PP.
That means you're going to have homozygous means 2
Capital P's.
2 capital P's, exactly, right? But can you put, when you make your like your gametes, do you put both of those P's into one gamete?
No.
No, right? Because what happens during meiosis?
Separate.
Exactly. The homologous chromosomes separate from each other during anaphase 1. So that's basically what he was calling the law of segregation. So he's saying that the alleles from the parent's genotype basically separate into separate gametes when forming the gametes. Okay? So that means that one parent can't contribute 2 P's. Each has to contribute only one single P to make a final double P genotype of a offspring, right? All right. So, for example, an individual with BB genotype, right? Is this heterozygous or homozygous? Heterozygous, right? So this law is basically saying that this capital B and the lowercase b won't go into the gamete, but that this one B will go into one gamete, and this other little b will go into a separate gamete, right? That's the law of segregation, okay? The next law he said was the law of dominance, which basically means that if there are 2 different alleles, right? Like, so for example, the big B and little b, one will be expressed and the other won't show at all. So the example is, if the individual's genotype is BB, capital B, lowercase b, they're only going to look like B, capital B. Okay? Does that make sense? This one, like, basically totally overshadows the other one, right? Okay, and then there's the law of independent assortment, and it kind of like, it's self-explanatory. So if they're different, like, genes, so not just the B gene, let's say, but also the gene for, like, the purple color, or, like, the shape, or all the different types of genes, they're not necessarily going to be in the same combination in the offsprings. So, like, suppose, like, this, let me just give you the example, the explanation first. So, alleles for different traits are inherited independently of each other. So, for example, if parents that are yellow and round, so this, right here, yellow and round is in one parent, and then the other parent is green and wrinkled in the other parent, right? So this is one parent, and this is another parent. The yellow and round two traits, even though they were in the original parent, they don't have to stay together, because you could get a yellow and wrinkled kid, and you could get a round and green kid, right? So these things don't have to be, so yellow and round don't get inherited together necessarily. They could all mix up and switch. Okay, so each different gene is inherited separately from each other. And how do we know, what causes that? What part of meiosis causes that to happen? What part of meiosis would make a yellow and, suppose yellow and round are on one parent's chromosome, right? Here's yellow, let's say, and here's round. What happens during meiosis that might cause them to be on different chromosomes then?
Synapses.
Synapses and the crossing over that occurs as a result of synapses during which phase does that happen at? Prophase 1, exactly. So because of that exchange of genetic information during prophase 1 of meiosis, that causes all these different traits to be like all mixed up, right, between the different homologous chromosomes. Good. All right. So everyone's with me so far? Yes. Okay. Good. Okay. Dominant. You guys know what dominant means. That's going to be the allele that refers to an allele, right? That is the capital letter, and it's going to overpower its corresponding lowercase letter one, right? Whoops. So, for example, T will be for tall, and then Y will be for yellow, right? The convention for this is to just use the capital letter for whatever that dominant trait is, and for the short one, you're not going to say like lowercase s, you're just going to take lowercase of the dominant trait, so T, right? So do you see how we didn't say S for the allele for this one? It has to be the same letter, and the letter is determined by the dominant trait, but then you just put a lowercase letter here, okay? And yellow, the same thing, right? So capital Y refers to the dominant trait, which is yellow, whereas lowercase y refers to green. You wouldn't put G instead, right? Okay. And then the dominant alleles, this is the law of dominance again, they cover up the recessive ones like entirely. So you're not going to see green or like, some weird like yellowish green. You're just going to see yellow. Okay? Even if you're heterozygous for that trait. Good? Okay, so let's start with this. If purple flowers are dominant over white flowers, what are the symbols for the alleles? So let me ask someone, let's see, Juliana, what do you think? If purple flowers are dominant over white flowers, what are the symbols for the alleles? So how do we determine the letter, first of all?
It's the capital letter of the dominant.
Right. So the dominant here is purple.
Purple.
So it would be
Capital P.
Capital P. And what would the allele for the white flowers be?
A lowercase.
Lowercase.
W.
Lowercase p. So like the letter for the same trait, because they're both the color of the flowers, the same. The trait is for the same trait. You always use the same letter, but for the recessive one, you use a lowercase of whatever the dominant letter was, right? So it's going to be capital P for purple and lowercase p for white. Okay? Okay. What's the phenotype for a plant that has a genotype of capital P, capital P? Mike. So what's a phenotype again?
Phenotype is like the like the one that doesn't show.
No, phenotype. That's you're thinking of recessive.
So is it the genotype, the thing that doesn't show? The phenotype is like it is what shows.
Yes.
Okay.
So what's the phenotype? Meaning the physical appearance of this gene, something that has a genotype of this. What's it going to look like?
It's going to be purple.
Okay, good. Thank you. Does that shows up purple for you guys, right?
Kind of looks pink.
Pinkish? Yeah. I don't know. Some people say it's pink too, but it's supposed to be purple. So, okay. What about capital P, lowercase p? What's the phenotype of this one going to be? Arna?
The phenotype phenotype is going to be purple.
Okay, that's also going to be purple, right? And then what about the phenotype for lowercase p, lowercase p? Hannah?
It's gonna be white.
Yes, exactly. That's going to be white, right? So how do we tell the difference between these two? Oops. Sorry. Wait a second. Hang on to that question for a second. Okay, so first of all, in Mendel, right, you guys have to know the difference between, in these classes, you have to know the difference between the parental generation, the F1 generation, and the F2 generation, right? The parental generation, so what Mendel did is he crossed all these flowers, right? So the first generation that he crossed were two true breeding plants, right? Do you guys remember what the definition true breeding means? Lidu, what does true breeding mean?
That means they're like pure purple or like pure white.
In terms of, what do you mean pure purple or pure white?
Like that's their not dominant, they're all purple genes.
Their genotype, right? So they're going to be homozygous, right? So
Yeah.
tell me the genotype of this one if this is true breeding.
That would be both uppercase P's.
Okay.
So 2 uppercase P's, 2 lowercase P's.
Okay. So 2 uppercase P's and 2 lowercase P's. This generation, homozygous dominant, homozygous recessive, that generation, that's known as the parental generation. This one right here. Okay? And then, so what Mendel did was he crossed these two parents, and then he got all purple offspring, right? And this is called the F1 generation. So that's like the first generation that he got, right? Do you guys, can you tell what their genotypes are? We know their phenotype is purple, but what do you think their genotypes are going to be? Kaylin, what do you think? Uppercase P and lowercase p, exactly, and that's why we're going to do a Punnett square to show why that happens, right? Do you guys know how to do a Punnett square? Has anyone done this before?
Yes.
Yeah.
All right, so it's no.
Yes.
Okay, some yes, some no. Okay, so we'll go through it just so you know. All right, so the parental generation. First, we take the parental generation, right? This was the purple one, which was homozygous, true breeding, capital P. This is the homozygous, true breeding, recessive, lowercase p, which was white, right? So the first question is, what alleles does each parent make? So Lidu, what was the genotype of this again?
2 capital P's.
Good. So what kind of alleles does it have? Does it have
Capital P's.
Good. So it can only contribute a capital P because that's all it has, right? So, and what is the law of segregation say? Mary, do you remember?
The alleles separate when they form the gametes.
Exactly. So what you're going to do is you're going to put on this box, for one parent, you're going to put on the top, you're going to put the separate alleles for that trait, one here and one here. And then for this one, right, we're going to separate the alleles and we're going to put one here and one here, right? George, can you tell me what alleles this one makes?
Lowercase p and lowercase p.
Right, so we're going to put a capital P here, and lowercase p here, and lowercase p here, right? P is like a really bad letter to do because they kind of look the same, but you guys can just differentiate between the capital P and the lowercase p, right? All right, so, and now what we're going to do is we're just going to bring this one down here, and then we're going to bring this one across here to get the ultimate genotype of what this offspring will be, right? And we're going to get the overall ratio, right? Excuse me. All right, so let's see. Anthony, what's going to be the result of the cross between this allele and this allele when we put them together? What should be in this box here?
Capital P, lowercase p.
Exactly, good. And what is that? What description is that called? If you have a well, first of all, what is capital P and lowercase p? Is that a phenotype or is it a genotype or is it an allele or what is it?
Genotype.
Well, one would be dominant because it's expressed over the other.
Yeah, so the capital P is going to be expressed over the lowercase p. But if I give you a combination of two letters, of the same letter. Is that, what is that? Is that a genotype? Is it a phenotype? Or is it an allele?
Genotype.
Genotype, exactly. So the genotype, right? So the offspring genotypes are going to be PP here. What's going to be here? And yeah, P is also a bad letter to choose for this because it's like end up saying PP a lot. So anyway, this one, what should go? Which one should go right here, Charlie? So we're going to bring this down. We're going to bring this across.
Capital P, lowercase p.
Okay, good. All right, do we see a trend here going? Anna, what's going to go here? Anna, are you there? All right, Aria, why don't you go ahead?
Lowercase p.
Okay, exactly. And then what about here? Olivia, is your mic working today?
Capital P, lowercase p.
All right, good. So these are all the genotypes, right? You guys all recognize that these are the genotypes? What are the phenotypes of all these then? Based on the law of dominance, what's the phenotype of all of these going to be?
The capital P.
The capital P we can't, that's not a phenotype. The capital P is an allele, but a phenotype is a physical description, right? So the phenotype purple is going to be purple, right? Are we happy? Yes? Too easy? All right, let's spice it up a little bit now. All right, so now we're going to take, so we recognize that all these ones right here, that's the F1 generation. This is the parent generation, this true breeding one and the true breeding one. The F1 generation is going to be those kids, right? The ones from the products from that last cross. So we know the genotypes of them, right? So what alleles does each parent make? Omar, what alleles does this one make? We know the genotypes of them because we just established them. What's the genotype of this one? Omar? The genotype.
PP, like capital P, lowercase p.
Okay, good. And what's the genotype of this one?
Wouldn't it also be capital P, lowercase p?
Exactly. So they're basically like twinsies, right? They're just like, and they're crossing with each other, right? And what alleles does this one have then that it can contribute? So we're going to separate the alleles, right? Because that's the law of segregation. So what should I put here and what should I put here?
The capital P and the lowercase p.
Good. I should put a capital P here and a lowercase p here. And the same thing goes for this one, because this is the same genotype, right? So it should all look like this, right? You guys all set that up okay? Okay, and now what do? Again, I bring one down from here, and I bring this one here. So I want to know what the genotypes of F2. F2 is the filial, like the second filial generation, the second like offspring generation. So these are going to be the F2 ones, right? These are the F1 ones, and the parents were the original white and purple one, right? Okay, so row, what's going to go right here?
Two capital P's.
Two capital P's. What about here?
Capital P and a lowercase p.
Okay, and it should go here too because those are the same, right? And what about right here?
Two lowercase p's.
Got two lowercase p's. Are we all good for this? Yes? Okay. And then I want to know what the genotype ratio of, like the ratio of each different genotype to each other. How many different genotypes do we have, first of all? Let me think. What am I missing? Sarah, how many different genotypes do we have here?
Two or four.
How many?
No, I'm in three. Sorry.
Three, right? So what's the ratio of how many of these we have to how many of these we have to how many of these we have?
1 to 2 to 1.
Exactly. Good. That's what I should. So if I'm asking for the genotype ratio of them, I would get 1 to 2 to 1. Now I want to know what the phenotypes of all these children are, right? Arsh, can you tell me what the phenotypes are here? What's this phenotype? Arsh, are you there? Are you unable to get muted? Unable to get unmuted? No?
Yeah, I don't think his mic is working.
Mic's not working. Okay. see, Kaylin, what do you think? What's this phenotype going to be? Purple, right? What's this one? What are these two going to be? Purple also, right? And what about this one? White. So, what is the phenotype ratio then of these children here? Let me ask someone else. Who have I not called on yet? I'm like trying to think of like all of you guys offhand, and then I'm having trouble here. Does anyone?
Can you hear me now?
Huh?
Can you guys hear me now? Alright.
Yes.
Okay.
All right. Let's see. Addie, what's the phenotype? 3 to 1, right? Okay. Is this like super easy? Yes? All right. So, question. If we have a purple flower, let's have a purple flower. Here it is, right? How will we know if it's homozygous capital P, or heterozygous capital P, lowercase p, right? Because they're both going to look the exact same because this one dominates over that no matter what, right? So I want you guys to tell me how I can determine this. Can I like cut it open and like figure out its genotype from that? Think about like what resources were available to Mendel at that time. Like all he could do, he couldn't like, analyze the DNA or the genes. So he would have to perform some sort of cross with some other individual of a known genotype in order to figure out what this one's genotype is, right? So, let's see. George, any idea of like what you could do, what you could cross this with to figure out whether it's capital P, or lowercase p, or capital P, lowercase p?
You could cross it with the white, like the all white one, the 2 lowercase p's, and
Exactly.
if it becomes purple, then you'll know that it's red.
Wait, what?
Well, you cross it with the white one, like the 2 lowercase p's.
Yes, and so tell me what's going to happen if I cross this one with the white one. What am I going to get?
You're gonna get 4 of the capital P, lowercase p ones.
All purple, right? They're all going to
Yeah,
look right?
the phenotype will all be purple.
Exactly. But if I cross this one with the white one, what will I get?
You'll get some of them purple, but then one of them will be, 2 of them will be white, and then 2 of them will be purple.
Exactly. So if I get any white ones at all from this cross of whatever this purple one is with a completely white one, that means that there must have been a lowercase like allele, right? Like a recessive allele in this original genotype, right? If I get them all purple, then I know that it's probably homozygous purple, right? So that's exactly what the answer that I wanted. And that process is called a test cross, which means that you're testing like an unknown genotype. Like we know the phenotype because you can see it, right, visibly, but we don't know. So a test cross is when you have a visibly dominant phenotype, right? But you don't know what the genotype is. So you cross it with with a homozygous recessive individual. In this case, which would have been lowercase p, which is white, right? And then if you see any individuals of the lowercase, like of the recessive phenotype from that cross, and there should be about 50% of them, then you know that the original parent was heterozygous. If you see 100% of the offspring that are all completely the dominant phenotype, you know that the original parent was probably homozygous dominant. Okay, so let's just look at what that cross would have looked like, right? So again, here are the unknowns, right? Here's the unknown flower, and it could be this, or it could be this, right? So if I cross these dominant alleles, right? This dominant homozygous dominant individual with just the test cross, right? I'll get purple, all purple, right? And then if I cross this one, the heterozygous, if it turns out to be heterozygous, then I'll know because this one, that'll be purple, right? That'll be white because 2 lowercase p's come together, right? This one's going to be what?
Purple.
Purple.
That one's going to be purple, and what's this one going to be? Again, lowercase, so it's going to be white, right? So by looking at the ratios of the offspring in your test from a test cross, you can determine an unknown dominant phenotype individual's genotype. Does that make sense? And are you understanding all the different terms that I'm using? So genotype is like the actual double lettered, it's the same letter, but it's double, you have one of each, because that's an actual individual. Whereas once they separate into the gametes, right? And each gamete has only a single allele of each given gene, right? All right, I think I'm done. No, oops, okay. So let's do an example one here, okay? And I think I need to go to normal, and then actually add in the answers because I don't have the answers here yet. So why doesn't someone can someone kind of talk me through this and tell me what I should do in the beginning? Who have I not called on yet? Have I? Let's see. Hannah, do you want to start us off here? So.
Can you repeat the question?
Yes.
Yeah.
Yeah. Do you want to read the question? Can you see the question?
Yeah. In pine trees, long needles are dominant over short needles. Cross a homozygous dominant plant with a plant that is heterozygous. What is the probability that the parents will produce offspring that have short needles? Give the genotypic and the phenotypic ratios.
Okay. So first of all, we're given that we have long needles are dominant over short needles. So what's the letter that I'm going to use to
L.
L, right? So the capital L is going to be so I'm going to put a capital L here, right? And oops, just kidding. And then I'm going to put a lowercase L, then I'm going to have a lowercase L for my other allele, right? And then what kind of parents am I given here? So here are my two alleles that I'm going to use. And then I want to cross a homozygous dominant plant. So that's going to go up here. Let's say that's homozygous dominant. What's the genotype look like for homozygous dominant?
It would be two capital L's.
I'm sorry. Yes, two capital L's, right? So I'm going to put the two capital L's, let's say up here. And then I'm going to cross it with a plant that is heterozygous. So what's this other parent over here's genotype going to look like?
One capital, one lowercase L.
Okay, good. And then what do I have to put here on the top of each boxes? So what happens to these alleles? So this is the genotype, right? But I actually want to I have to separate the alleles into gametes, right, when I make the cross. So what do I have to put here and here?
So you just put an L over each box.
So what kind of L?
Capital L.
Good, okay, so I have a capital L here and here. And then over here, what should I put over here?
A capital L and then a lowercase L.
A capital L and then a lowercase L, good. Alright, oops. And then a lowercase L. Alright, you guys are all with me, right? Alright, okay. And then, now what do I do? What are the results then, Hannah? Can you tell me? So what's going to go here?
Two capital L's.
Okay, and I'm going to put that right there. What about here?
And then the next box, two capital L's.
Okay, so I'm going to put that right here. Alright, what about here?
And then a capital and a lowercase L.
Okay, and that's going to go right here. And what's going to go here?
And then another capital and lowercase L.
And that's going to go here. Okay. So tell me about their genotypic. Tell me about the genotype. How many different genotypes are present in these offspring?
Two.
There are two different ones. What are those two different ones?
Two capital L's and then one lowercase, and then two of them were one lowercase, one capital.
Exactly. So what's the ratio? What's the ratio of one genotype to another genotype?
1-to-1.
Just 1-to-1. You could say 2-to-2 if you wanted, right? But yeah, it's basically just 1-to-1, right? And what's the probability then? Oh, and what about the phenotypic ratios? How many different phenotypes do I have here among these offspring? One, right? So what's the phenotypic ratio?
Just 1-to-1.
We only have one phenotype, so it's gonna be pretty much like 4, because they're all long needles. 4 to 0. Okay. And then tell me, what's the probability that the parents will produce offspring that have short needles?
0%.
0%. Excellent. All right. So you guys All right. Who wants to do the next one? Any volunteers, victims? Let's see who I have here. Who's there? Some of you guys have your cameras off, so I can't even see you. But I want to pick on you. Let's see. I think I should do this one. Sarah, why don't you do this one? Can you read it out loud first, and then?
Okay, yeah. In cats, a striped coat is dominant over a solid coat. Cross a cat with the solid coat with a cat that is heterozygous for a striped coat. What is the probability that the parents will produce offspring with a solid coat? Give the genetic, or the genotypic and phenotypic ratios.
Okay. So first of all, like, what alleles are we? What letter are we going to use for the alleles?
It's uppercase and lowercase s.
Yeah, this is like a really bad example because like this is S and this is S too, so that's so stupid. But yeah, so capital S is going to refer to the what? That capital S is going to be the
Striped coat.
striped, right? And then the other allele is going to be solid. Is going to be lowercase s. Okay. All right. So we're going to cross a cat with solid coat. So if it has a solid coat, that means it's this lowercase s. That means we know the phenotype. We know the phenotype, but we also know the genotype. Why?
Because it says it's heterozygous.
Wait a minute. It says cross a cat with a solid coat.
Because it wouldn't if there was a dominant trait, it would show the dominant.
Right. So if it's expressing or if it's showing the recessive trait, we know the recessive phenotype. We know it's definitely going to be homozygous recessive because that's the only way the recessive trait would ever come out, right? So we know that one of the parent is like little s, right? Let's actually put that up here just for consistency because that's the first parent that we're going to use. These are just the alleles, right? So I just took the alleles and put them over here. But capital S is striped. This is solid. So we have a solid coat cat, and then we're crossing it for one that's heterozygous for a striped coat. So that phenotype, the genotype of that one is going to be what?
Uppercase S and lowercase s.
All right. And so what alleles can this one donate to the Punnett square?
Lowercase s and lowercase s.
Just a lowercase s and a lowercase s, right? All right. Sorry. Here, we'll put that here, and then we'll kind of paste it. We'll put this one over here. All right. What about the other one? This one's gonna
Uppercase and lowercase s.
Okay. So uppercase s here, and then lowercase s is gonna go right here. Okay. And then. So what is the probability that the parents will produce offspring with a solid coat? So let's do all the crosses and see what happens. So what's this cross going to result in?
Capital and lowercase s.
Okay. All right. What about this one right here?
Bottom left, it's SS.
Okay. Lowercase. Okay. And what about over here?
Capital S, lowercase s.
All right. That one's going to go. And what about this one down here?
Lowercase and lowercase s.
All right, so that one's going to go over here. So now, let's see, what's the probability that the parents will produce offspring with a solid coat? So which one was solid again? The phenotype?
The capital S.
Careful. That's what...
Oh, solid, yeah, sorry, lowercase.
Right, so what's the probability? So these are going to all be striped, right? These are going to be solid, so the probability is...
It's like 50%.
50%, exactly. And what are the genotypic and phenotypic ratios? So the genotypic ratio is...
1 and 1.
1 to 1, right? And then what's the phenotypic ratio?
1 to 1.
Also 1 to 1. Or you could say 2 to 2, and that would be fine too. Alright, everyone's with me? You want to do one more or no? Are we ready to just go and do the homework?
Homework.
You want to do one more? Yes? No? No? You don't need to do another one?
I think we're good.
Alright. Okay, I'm going to stop sharing my screen. Alright, so you guys all understand this? Yes, and you can figure out what the genotypic phenotypic ratios. You know what true breeding means. You know what a test cross means, right? And let me just say, one thing that's kind of weird about plants is that you can let them self-pollinate themselves, which means that suppose I say, okay, I have parent generation. What were the genotypes of of Mendel's parent generation again? Mike, do you remember the parents that Mendel used in the very first cross that he did? What were their genotypes? two parents that he chose were both true breeding. One was dominant, one was recessive. So it means that one was homozygous dominant, one was homozygous recessive, right?
Oh, wait, so it would be like
Two capital letters.
two would be like capital letters and two would be lowercase letters, like
Yeah, one would be two capital letters and one would be two lowercase letters, right?
Yeah.
So we take that those original parents, and what's the genotype of all their kids that they're gonna have?
Wait, can you say that question again?
What's the genotype gonna be of all the offspring that they produce?
Would it be like what the capital?
That's
Like,
that's the phenotype. They're all gonna demonstrate the capital alleles phenotype. But I'm asking for the genotype. What's their actual? So if I have a parent that only can give me capital letters, right? And they donate one capital letter, and then I have another parent who can only donate lowercase letters. They're gonna donate a lowercase letter. So what's the kid gonna have?
a capital and a lowercase?
Capital and a lowercase. That's called heterozygous, right? So they're all gonna be heterozygous for that trait.
Yeah.
And what generation is that? All those heterozygous kids. What generation is that one called?
Wait, would it be second generation?
But in our terminology that we're using today, what was it called?
F1.
Offspring?
Mm-hmm.
F1.
It was called F1,
Oh.
right? So F1 generation, and then some of the problems on your answer, right, might say, what happens when that generation is allowed to self-pollinate, which means that they're basically just being crossed with someone of the same exact genotype, right? Okay? Are you guys with me here? Yeah? So the term self-pollinate means that you're just being crossed with someone with the same, or with yourself, right? With the same exact genotype that you have. So if I let the F1 generation self-pollinate, that means I'm crossing capital P, lowercase p with capital P, lowercase p, right? Suppose I let a true breeding plant self-pollinate. What's going to be the results? True breeding means that they have just two of the same alleles. Is there ever going to be an introduction of any other allele if this one thing is self-pollinating?
No.
No, right? So if I have a white flower and I let it self-pollinate, all the kids are going to be white, right? If I let a like a true breeding purple flower, meaning a homozygous dominant purple flower self-pollinate, all the kids are going to be homozygous dominant purple, right? Because there's no other alleles being introduced. Are you guys good with this? Yeah? Do you feel okay? Any questions? No? Alright, so go ahead and you're going to have to set up the Punnett squares on a lot of these, on a lot of the homework questions. It's just like, it's practice. It's good practice. How many alleles for a given trait go into each gamete?
One.
One, right? So if I ask for what would a gamete look like for like, the purple trait, or the color, the plant color, the flower color trait, what would you look for? Would you look for 2 P's or 1 P?
One.
1 P.
1 P. But if I ask you for a genotype,
Two.
what would you look for?
2.
2 P's.
You need 2 of the same letter, right? So 2 P's. But what if I ask you for a phenotype? Would it be expressed in letters?
No.
No. If I ask you for a phenotype, then you express it in actual, like, physical descriptions, like, purple, right? Or like wrinkled or round or yellow or something like that, right? Okay. I think that you guys are all okay with this. So I'm going to let you guys go ahead and work on that, the worksheet, and then it will be due on Monday, and then on Monday we'll start talking, we're going to add in, we're going to spice it up and add more traits, at the same time, and we're going to do like a dihybrid cross, which is, instead of 4 boxes, it's 16 boxes. So we really need to make sure you guys know, your stuff, right? Fun? Okay. All right, I will post your scores for your quiz from yesterday in a few minutes. So have a great weekend. Enjoy yourselves. Go outside. You can go to parks again. Did you hear that? Starting May 1st, you can actually go to parks.
There's a stick bug!
stress and enjoy the weather.